in pursuit of health and wisdom. Sapio with Buck Joffrey.
Welcome back to Sapio with Buck Joffrey. Today, my guest is Dr. Diogo Barardo. He is currently the Director of Research and Development at NOVOS, which is a leading company in longevity supplements. Dr. Barardo earned his PhD in biochemistry from the National University of Singapore, where he later worked as a postdoctoral research fellow at the renowned Center for Health Longevity under Professor Brian Kennedy. His research has been widely recognized, with studies cited in over 1,500 research papers. One of his most significant contributions to the field is the creation of DrugAge, which is the world's largest database of longevity-enhancing compounds across multiple species. He's also known for authoring the first paper on using machine learning to predict anti-aging compounds. His expertise spans from working with simple organisms like C. elegans to mice and humans, so gives them a comprehensive understanding of the aging process across different species. Welcome to the program.
Thank you so much. It's a pleasure to be here. Thank you.
Yeah, so this interesting stuff. So why don't we start with this, Diogo, is tell us about NOVOS and like sort of how they're different from other companies in the space.
All right. So NOVOS or NOVOS Labs, it's the largest direct-to-consumer company that provides longevity solutions. I think the main distinguishing factor for NOVOS is that we take science very seriously, and we are evidence-based, and we invest a lot, for a startup especially, we invest a lot in R&D, that I'm the director of, to validate our compounds, our solutions, and also to come up with new products to help people in their longevity journey.
Right. So you're a biochemist, and you specialize in longevity research. So when you're looking at the question of aging, how are you defining it from a biological perspective? Maybe you can kind of talk about the difference between, biological aging and epigenetic aging and why you might have chosen one over the other.
All right, sure. So I think first, starting from the beginning, what is biological aging? And so if I want to know how old you are, I can just go to your wallet and, check your passport or check your citizenship card. That's not very informative to the state of your health. We do know that older people, as you know, suffer, a lot more disease. They have a highest risk of all-cause mortality, but that's not really enough to distinguish people that, for example, have relatively the same age and the same age group. So biological age is this concept that there are signatures within people, even in the same chronological age group, that allows us to distinguish them between people that are at increased risk and decreased risk of all the major diseases that are associated with age. Degenerative disease, cancer, and so on. And so the way we do it more technically is that I think a good example is, for example, we all know people that if we look at them, it's like, he looks much older than he is. And we also know the reverse. There are people that look much younger than they actually are, and then we're surprised, oh, wow, okay, I didn't know you were that old. You look much younger. Yeah. So we basically convert this kind of notion in the technical terms using AI. We convert this and we apply it to other types of data. So, for example, if it's based on facial recognition and you look, your face looks like someone much younger, your facial age or facial biological age is that of someone younger. If it's applied to epigenetic data, so I don't know if maybe I should explain to your listeners what is epigenetics, maybe very briefly.
Yeah, maybe just briefly. I think the difference between biological aging and epigenetic aging is probably useful.
Okay, so epigenetic aging is one way of measuring biological aging, one very specific way. So you can measure biological age in any way you want, that you find some kind of signature that allows you to distinguish individuals across their lifespan. So if you, in certain part of your body, in any way you want, even aesthetics, you resemble someone younger, you would be biologically younger according to that parameter. If you resemble someone older, you would be biologically older.
Right. So, but in this case, with biological age, you're looking at typically you're looking at markers, like you're looking at laboratory studies. Just like you can look at somebody on the outside and say, boy, you look 10 years younger. You can look at their insides by looking at their lab results and say, boy, you look 10 years younger on the inside by what your labs are showing. Is that right?
Exactly. So there are even, the normal blood counts that any, GP prescribes to just, in a normal annual checkup. There are actually blood-based biological aging clocks built on this data set. And much more important is that we just don't want, okay, you look younger and older, but what can you actually use this for? Well, it's very informative. So basically, if you look older, you have increased risk of mortality of pretty much everything. You have increased risk of frailty. You have increased risk of multimorbidity. It has a very strong implication for your health at the personal level.
Let me ask you this. So we were distinguishing a little bit between, we were kind of hinting at the distinguishing between biological aging and epigenetic aging. And at least the way I think about it is, biological aging is using a lot of serum markers, right? You're using a bunch of blood tests that you, maybe you're looking at inflammatory markers or any number of markers. Whereas epigenetic aging, you're really looking at the effects on the epigenome, part of the genetic material. So when you guys are looking at that idea of aging, are you using more genetic information? Or are you using more of the biomarkers?
Okay, just maybe to try to clarify better, epigenetic aging is one measure of biological aging. Sometimes people, they don't really, also is a matter of just sometimes even marketing, is they just say biological aging equal biological age to everything. But as I said, biological aging is the concept that there is another age that is not the chronological age. Exactly. But epigenetic is just a type of it.
Okay.
So in epigenetics, we don't measure your genome per se. We measure how accessible it is to the cell. So the genome, as we know, is the manual of instructions of the cell, of all the possible things that the cell can possibly need to do or is able to do. And not all the pages of this manual are available to be read. And that's a good thing. Right. Because, for example, all the cells on your body, they have the same genome, but they are very, very different functionally. A adipose tissue cell is very different from a skin cell, which is very different from a heart cell. So you want to maintain this, we call it differentiation, but we want to maintain this specificity to the cell type. And the epigenome is the thing that controls which pages of the book can you read. Right. And it gets very laxed with age. So certain pages that you shouldn't be able to read, now you are. And certain pages that you should be able to read, now you cannot read them anymore as a cell. And this is what we measure.
Okay, got it. So yeah, I mean, I think the confusion sometimes is, when a lot of people have read David Sinclair's work, and he makes a very clear distinction of what epigenetic age is in his information theory of aging, right? Which is, DNA methylations or any other chemical modifications of the epigenome. And I think what you're saying is, yeah, that's right. That's part of it. And then you're going to add that to any number of other blood markers that you're seeing and create a complete picture, which is biological age. Is that right?
I think what you're trying to allude to is that biological age should be somewhat moralistic, since that's just one aspect of it.
Right.
Yes. So you can have these different measures of biological age, and then you can combine them into, this moralistic measure. Yes. So that's actually now where we are moving towards in science. Like the latest research is using these different types of data from different sources of the human body to then come up with this composite biological age.
Got it. Let's talk about the drug age database, because I think that obviously is a big contribution. Tell us a little bit about that. How does that database, how is that generated?
Okay, so that basically is a result of my master's. So I was in the University of Liverpool at the time in an aging lab. When I compiled it, I was actually we had this huge Excel spreadsheet with all the studies, and I was compiling by myself. So the drug age is a database that has all the compounds that have ever been tested in model organisms and shown to extend lifespan. We do this because obviously we cannot test in humans, since it would take decades to have a readout and waiting for people to die. When I was working on it, there was no such resource available. And for me, it was, okay, I want to join this field, I want to contribute, but I don't even know what my predecessors have done. So how can I help this field? And that's the result of it. So then my supervisor turned it into a database online, and now it's freely available. Anyone can just Google drug age, and you can check your favorite compounds to see, namely supplements, ingredients, and you can see there what are the studies that have been done in model organisms showing that it works or not.
So what are some of the most promising compounds in your opinion? Listen, right now there's so many supplements, right? And I was looking at the ingredients in your NovoScore, for example, and I was asking myself things like, well, there's no NAD precursor here, or there's no spermidine. There's not, there's so many supplements. I guess, what are some of the most promising compounds for longevity? And maybe a bigger question is, how do you determine which ones warrant further research?
Okay, so that's a great question. I guess that's the question. So maybe I'll start with that one, the approach that we take. So we want to be evidence-based as much as possible. And because we are in the field of dietary supplements, safety is a big consideration. Because obviously, if you have a very serious condition and you take a pharmaceutical drug, you're willing to have a certain trade-off between fixing your condition and, safety, right? As we know. But in supplements, I would say that there is no such trade-off. Like, you want things that basically work without side effects. So the first thing that we do is, are these supplements generally regarded as safe? Even that label is not enough. We also want to know what are very long-term studies at relatively high dosage. What are the side effects, if any? Is this compound a natural compound in the human body? Because usually compounds that are present in the human body, they are integrated in our metabolic pathways, so they are not xenobiotic, so they are not some external alien chemical in our body that has to be processed. So these are just the first phase. Then the second is, do we actually have evidence that extends lifespan in any organism or not? Because if it doesn't, maybe it's not going to be enough to move the needle. So we need a certain effect size, is what we call. So we need to see, let's say if we are working with worms, we need to see at least 20% lifespan extension, because we feel that's a good effect size, that the data is not noise and is not due to some specific laboratory conditions. Okay, but then with this we show the supplement is safe, so the toxicity profile is very good. It works in longevity in model organisms, but what about humans? So then what we want to see is that if for a certain supplement, we actually have readouts in humans. Obviously, we're not going to have longevity, but we have, or very rarely, there's some that we might have in epidemiological data that decrease all-cause mortality, but most of them, there is no such thing. So what we want to see is that, can we actually see beneficial effects on health? Namely, can we see beneficial effects on health span, or some measure of health span? And then if all of these point in the same direction, then we go to more considerations of the product line itself, which is, can we reach, can this be deliverable? Is this bioavailable? What is the optimal quantity, and so on?
Talk about, how do you determine, once you put together your formula of whatever you want in your supplement, how are you assessing the efficacy of the intervention?
Right. So we are a startup. So obviously, we're not some big pharma that can just spend millions of dollars in a randomized clinical control trials. Having said that, we do have, as of now, we have in vitro studies. We just published one actually in a peer-reviewed publication. We have studies for lifespan and frailty in mice. We also have just now a preprint that we show that NovoScore extends mouse lifespan by 18%, which is better than just some invertebrate model, which is relatively easier to have that effect. And we also have underway human clinical control trials. So it's just a matter of time for us to see if our formula lives up to our hopes or not.
You had a study in skin. There was a skin cell study, and I think that where you showed significance of targeting multiple aging processes simultaneously. Can you talk a little bit about that?
Is this because, are you referring to the case study in our website or actually the peer review study?
I think it's a peer review study on
Okay.
Skin cell studies. Yeah.
Okay, so in that study, basically, we tested NovoScore, our full formulation, in some subsets of it.
So that's your core supplement, right? That's the main supplement from Novos for longevity, NovoScore. So yeah, go ahead.
Yeah, that's our flagship product. It's also the first one ever on the market. And right now we only have 2 more because it's relatively hard to create products with this kind of standard.
Yeah.
And so regarding NovoScore, so on that study we had NovoScore in skin cells, and we also had subsets of NovoScore. And basically what that shows is that none of the subsets of NovoScore has any toxicity. They are also protective against DNA damage. So we subjected the cells to these very bad chemicals that basically they break apart DNA. And they are very protective. And NovoScore has slightly less or about the same protection, but at a much lower dosage than the subsets. So that seems to indicate that there is some kind of synergy between the components.
Can I walk through the NovoScore supplements with you? And I know there's a number of them in here, but just a few words on each would be useful. Alpha ketoglutarate. What's the benefit of alpha ketoglutarate?
So alpha ketoglutarate, it got to the spotlight based on the study from my previous supervisor when he was still in America, leading the Buck Institute. Uh-huh. And basically, it is shown that it extends lifespan in mice, and it might also improve the frailty index. That was when it became to the spotlight, and now more studies are underway. The mode of action is not clear, to be honest. We only know that it's good for you, but with the chemical, because the alpha ketoglutarate is relatively abundant molecule in the human body, and so it's hard to pinpoint what is the specific mechanism of it. There are several.
So there's a few different cellular senescence drugs that target cellular senescence. Rhodiola, rosea, and the other one is fisetin. There's an enormous number of cellular senescent targeting molecules. Why these two?
Okay, first fisetin. Fisetin seems to have a senolytic action. So senescent cells are aged cells, and they have some function in your body. We know that. They can be beneficial, but eventually it's also shown that we accumulate too many with age. So they basically are this suboptimal, sometimes totally non-functional tissue in the human body. So they occupy space, they take resources, but they don't do much of the functions that they should do for the tissue. So a senolytic drug is a drug that selectively induces the killing, or in this case, the suicide or killing of these senescent cells while not touching the healthy cells. So fisetin is one of such compounds.
Right. There's multiple compounds, sort of, again, just looking in here, you have L-theanine, hyaluronic acid, all targeting cellular senescence. I guess one of the questions, in part, it's a selfish question because I find myself on multiple multiple supplements targeting cellular senescence, including fisetin, including like spermidine, for example. And I guess as somebody who designs these supplements, I'm curious on sort of like, are you looking at this? I mean, what's the point of having like 4 or 5 drugs that all 4 or 5 compounds that all target cellular senescence? Are they targeting it in different ways, or what's the idea?
That's a great question, and it's quite complex. So first of all is that a lot of people do some kind of senescence assay in cells just because, well, that's what people have to try in vitro in the longevity field. They differ a lot in which molecules are very reproducible in this kind of assays, and a lot of molecules they were tested once and they might have some beneficial effect for senescent cells, but that was it. So for example, fisetin is widely known to be a senolytic, but not without some papers saying actually it isn't. You mentioned spermidine. Spermidine is a very interesting compound. It's actually a great example of for the approach that NOVOS takes, because we had an entire product in the product line that the main theme was be a great source of spermidine. And I had to withdraw that idea from as a product. So the product turned out to then now be Vital, which is a shoe that we have in NOVOS. But the theme of that Vital initially was being a great source of spermidine. The reason that I removed it is because I was not convinced at all by the human data.
Really?
So it seems to work very well in mammals, in mice, when they test it, both IV and orally. But then in humans, even the people that, their kind of academic career depends on spermidine working, they show that is not seen in the human blood after oral intake. So it's not really ultra convincing data, even though it works really, really well in cells and in mice.
Tell me about lithium. I've heard a little bit about this, but there is a microdose of lithium in the core product.
Yes, so lithium is used is FDA approved as a drug at much higher dosages, more than 100x the dose we use. That's why we call it microdose. And so at that dosage is used for the mental disease. It's
it's schizophrenia, I think, right?
Exactly.
Yeah.
And in those dosages and even higher, it can have some side effects. What we are using is a microdose of lithium that has absolutely no side effects. So this is used, so there are certain parts of the world, for example, that the normal concentration of lithium in the water supply is as much as we are putting in the supplement, or even slightly more. And there is some research showing that, for example, people in these particular areas, they tend to have slightly longer lifespan. Huh. Of course, this level of evidence is just associative and, epidemiology. So then in terms of human clinical control trials, microdose of lithium is now being tested only now. There are maybe like 2 or 3 studies, and it seems to have still relatively small sample size, but it seems to have a benefit for cognitive decline. In terms of animal science, is known that lithium extends lifespan, for example, in flies, and the target seems to be relatively well known. So we know the mode of action. It's very nice to know that. And it seems to be one of these things that it's been overlooked for a really long time.
Sure, that makes sense. There's a number of ones here that I would love to talk to, but in the interest of time, I want to ask you about some things that are not included. So, for example, in the longevity space, the NRs, all of the NAD boosters, basically, obviously going back to David Sinclair made a big push, I think, for the idea of boosting NAD plus levels, through precursors like NMN or through NR. Obviously, nothing made the list here. Curious why.
So we do sell NMN. We call it NOVOS Boost. We sell as a standalone ingredient. So we do have overall a positive opinion on it. It didn't make any of these lists for 2 reasons. It didn't make NovoScore because the research was still in its infancy when NovoScore was created. And it didn't make NOVOS Vital because we already have Boost. And even though NAD precursors are promising, they are a bit overhyped.
Yeah. No, I think at least in mice data, I think it does seem like in terms of the actual survival. What was it? The multi institution study. I can't remember the name of that.
ITP.
The ITP didn't show any actually benefit to that. And I guess along the same lines, resveratrol, similar reasoning there.
Yeah, exactly. So exactly the same. And for example, NAD is curious because it seems, so it's now being tested in clinical control trials for pregnancy.
Yeah.
And it seems to help with obese mice. So what I understand from this is that if your body is under a lot of biological stress, seems to be really efficacious. If it's not, if you're a couch potato, it's going to be great for you. You probably should take one. If you're not and you're an athlete, I don't know how much benefit you can get from it. It's an open question. There are no bad things about it, in a way. So you can just, take it and see for yourself. But yeah, that's my personal opinion.
You talking about resveratrol or NMN?
NAD precursors in general.
NAD precursor. What about resveratrol?
Resveratrol is maybe the most controversial longevity ingredient.
Yeah.
Long history, full of controversy. I don't know how much of it you are aware.
Yeah, the French paradox and all that stuff that got in. And then obviously Sinclair's study with resveratrol, but the question of what he was calling aged mice was a little confusing there, I guess. But tell us a little bit about that.
There is also, I think there is maybe, I don't know if you've ever heard of Surtees Pharmaceutical.
Uh-huh.
So it was the first big company that actually, a spin-off from David Sinclair, that actually was bought for literally hundreds of millions, I don't know the quantity, by Big Pharma. And then the project, the basically was canceled. So after that huge investment. So it's exactly. So I don't want to go deep into it. It's really controversial.
Yeah, okay.
So there are basically two of the leading experts in the world, which is David Sinclair and the other one being Matt Kuberline. They contest, they go head-to-head saying David Sinclair that resveratrol really targets, is really an anti-longevity compound with a very well-defined mode of action. And Matt Kuberline, plus I would say at this point, maybe the majority of the field contest that result and say that is due to, experimental artifacts.
Yeah.
Yeah. So it doesn't inspire a high level of confidence.
Sure. Yeah, that makes sense. I noticed on here there's a few drugs, and I this is an area that's actually kind of interesting for me, is various drugs that affect the mitochondria. So mitochondrial dysfunction, I guess, maybe in some cases, I guess, getting rid of senescent mitochondria or dysfunctional mitochondria and promoting the growth of new mitochondria. In terms of longevity in general, how big of a target is that?
It's quite big.
Uh-huh.
It's it's basically mitochondria. We always see it, and they are always introduced as the power plant of the cell because they produce energy and so on.
Yeah.
Or the energy is produced there. But they are much more than that. They are an organelle, and they live up to that name. So they have from signaling molecules to we also think the mitochondria, as we see in the textbook with that specific shape, like that almost rod shape.
Right.
But they are actually, it's basically a network of organelles, and they are fusing, and actually they suffer fission, so they get separated very, very fast to just respond to the micro environment around that cell and the compounds available. So they are really dynamic organelles, and we do know that they get, let's call it inefficient or even nocive to the rest of the cell with age.
Yeah.
And so the idea is that if we can improve, namely the big target now, even from Big Pharma, is mitophagy, which is taking all these mitochondria that are dysfunctional and recycle them to build new mitochondria. So they are trying to activate this pathway that does this recycling.
There's, again, you go into, we just talked about before how there's all these cellular senescent targeting drugs, and how do you pick one? For mitochondrial health, I've been taking like CoQ10, how do you determine which drugs to use? I think here you've got magnesium, but I didn't necessarily think of magnesium necessarily just as a pure mitochondrial, a mitophagy drug. You've got malate, I guess glucosamine. How do you choose? How did you come to those instead of, say, some of the ones that people talk about more often? Like, specifically CoQ10 is one that a lot of people talk about when it comes to mitochondrial dysfunction.
Yeah, so I give mitophagy is like the new trendy pathway related to mitochondria as an example. But neither of the compounds that we have in our product line, I would say, and also neither CoQ10 acts through that pathway. So usually they tend to be more the classical things that manipulate mitochondria in a pro-longevity manner are antioxidants. And so CoQ10 is one of them. It's actually one compound that I do have a positive opinion on, especially seems to be in some studies, it seems to interact very well with selenium. These are epidemiological studies. The problem with antioxidants in general is that for a healthy person, they might have a downside in the long term, obviously, which is, for example, it is known that if you exercise, the stress response that is beneficial, like the way that your body collects benefit from exercising is actually by getting the stress signal that basically when not kills you makes you stronger. Yeah. So if you take, for example, a very strong antioxidant, this is been replicated in multiple organisms, including humans, several times with different compounds, is that you block the signaling pathway, so you cannot collect the benefits of exercise. So there is a time and place for taking antioxidants and certain conditions and maybe some health states, but in general, and they are classical because they come from this theory of aging, which was the free radical theory of aging, that the entire aging process was created by these free radical molecules. So these, which the antioxidants fix in theory.
Yeah, yeah. Just along the mitochondrial side, there's a couple other ones I thought I'd just ask you about because they're all over in social media and stuff. And some of the influencers talk about them. For example, urolithin A. What's your take on urolithin A? Because that's it's a big influencer drug.
Yeah, so urolithin A actually is one of those very few compounds that does act through mitophagy. So that looks very promising. It's a very interesting compound because it's relatively new. We knew of its existence for a while. It's a natural compound, but it's relatively new in the aging field. I actually had some pleasure to analyze data from a study that was never published yet. It's not published yet, of urolithin in model organisms from Brian Kennedy's lab. And basically, it does seem to work at least in a few model organisms, but not all. By work, I mean extend lifespan. And the only downside I would say is that a lot of the studies in humans, if not all, come from the same lab, which is the lab from which the spin-off that sells urolithin A comes from.
Right. Again, it's a challenge, right? And that's part of the challenge in the space.
Yeah.
The other one that I've heard about, and I'm just curious on your thought. It's similar to CoQ10, but it's I guess it's a MitoQ or mitoquinol. Do you know this one?
Yeah.
Tell me about that one.
So that I would say also comes from that time. People were very excited about the free radical theory of aging. So if I'm not mistaken, it's been years since I read this research, but if I'm not mistaken, is a compound that specifically has bioavailability at the mitochondrial level, so you can actually reach not only the cell but the mitochondria. And then there, it is basically an antioxidant.
Yeah.
So it seems to be quite powerful. I don't know any studies, for example, in mice showing it extends lifespan, for example. Yeah. So I think it's still, maybe it's from that era of the free radical theory of aging.
When you talk about the NOVOS, what's the standard dosing like with the core? Is it you just take like 2 pills a day or something like that? How is that?
No, it's not in a pill form. It's a sake. I don't know what to say in English.
Okay. Like a powder form kind of thing?
Exactly. You pour it in your drink. You just, you move the liquid around, water or a juice or whatever you prefer with a spoon, and then you just
dilute. Is it like a once a day thing or twice a day thing?
Yeah, exactly. Once a day.
Once a day. So I'm curious on the dosing. Like this is the other thing that I find very, very enigmatic about supplements, because even when I, for example, you look at some of the NAD precursors, right, and the dosing from the companies themselves a lot of times is a lot lower than anything that in the studies that actually shows any benefit. So, for example, like if you look at NMN products, typically, I mean, the dosage that they're typically looking at is maybe 250 to 500 milligrams max per day, where my understanding is that, the dosing for any sort of therapeutic benefit is probably a little bit higher. So, I mean, in some respects, it's like, what's the point of even taking it if you're not taking therapeutic levels? How do you determine for your supplements, what therapeutic levels should be?
Okay, so I would say that there are two types of studies that, when a compound comes out, we're talking about human clinical control trials, or at least clinical trials if they are not control, which is some studies that are just academic in nature, so no conflict of interest, no sponsorship from the company, and they are studies sponsored by the company. I can tell you that both of them, because there's always something to lose. On the academic side, you are investing, time and money, and to kind of show that this thing works, that you believe it works. You got a grant for it. It's a KPI in your profession, in your career. And on the company side, you're investing money by sponsoring the study to prove that your thing works. So because of this vested interest, what happens is that instead of giving to the dosage that they are pretty confident is going to work, they give to a dosage that is safe, but it should really, really work. Like the signal should be clear that like we take no risks here. Right. So in general, this is what happens, that the dosage in clinical trials tend to be higher than they would be otherwise.
Got it. So, so the other thing that I know that you've been working with and is artificial intelligence and some other omics. Maybe you can explain kind of what that is and how you're applying it. How do you apply those technologies beneficially for learning more about what potential supplements will be useful for longevity?
Yeah. So when I described NOVOS initially, I described as a longevity solutions. And the reason is because we are not just a supplement company. We also have several things in our offer. So for example, for free, you have an app that you have a validated aging clock that is based on the questionnaire and totally free. You have a longevity questionnaire to help in your journey. What are the kind of limiting factors and so on. So we are offering solutions not only on the supplement side, but also on to track aging. So that so basically you want to create a loop that you measure where you are. Are you really interested? Do you really need supplementation? Because maybe you just need to get the basics right. Maybe you just to fix your sleep, stop drinking a lot, and just follow grandmother's advice. Right. But maybe that's not true. Maybe you already do all of that, and somehow you did an aging test. We also have an epigenetic aging test that we sell, and you are biologically older, or at least you're not as young as you thought you would be. And so then, okay, you're getting all the basics right. You need to add something up. And then we have the supplements. So we want to create this feedback loop from you can assess, you have solutions, both free and also, supplements that you need to pay for, obviously. And then you assess again. So you don't need to trust us. You need to trust us that we did the homework and we are giving you a potential solution, but then you can see it for yourself if it works for your individual case. And this is how we use AI. So in this case, my expertise is what's called aging clocks, as we talked about, but in this case is AI powered. So what happens is, for example, epigenetics aging clocks, they take about 1 million data points. So 1 million of these pages in the genome with instructions. How readable are they? Are they totally unreadable or they are readable? So that's a lot of data, and you cannot give that to a human.
And that's data that's genetic data, epigenetic data. It's blood markers, inflammation, all that stuff combined together, right? Is that?
Yeah, exactly. So multi-omics is exactly that. So there are different. Omics is basically a way using a very well-defined, let's say, category of molecules. So for example, lipidomics is I take a look at only the fats in your body. And metabolomics, I take a look at metabolites in general, and so on. Transcriptomics to the transcription, so the molecules that create the proteins eventually on your body. Proteomics, I look at the proteins and so on. So if you combine all of these ways of looking at the human body, the human biology, you have a more robust signal of aging, much more holistic. And the thing is that these are millions of data points. So you have no other way than to use AI. There is just no other way.
So one of the things I always wonder about these clocks too, and let's just take the epigenetic part out, but like if you use blood biomarkers. So I'm a guy who's like, I take a lot of supplements. You know, I am like, for example, I probably overtreat my cholesterol to artificially suppress my ApoB and, you know, all these kinds of other things. If I do an aging clock, it's going to end up showing a pretty good result, but a lot of it is pharmacologically, you know, it's masked by all the pharmacology that I've already done in my body. So the question is that a legitimate aging clock anymore?
So that's a great thing. So in your case, if you have LDL and, for example, very low levels of LDL or ApoB, as we know, with the PCSK9 inhibitors usually.
Right. I take one of those.
Exactly. So they do have, I think there is, at least in my opinion, there is a lot of convincing evidence that lower levels are better. And so I do think that even if you're using artificial mean or a pharmaceutical mean to reach those values is beneficial for you in the long run, assuming that you don't have any side effects, obviously, from the drugs.
Right.
Then there are things that, for example, there are certain things that aging clocks can be manipulated, and then they're not an aging clock anymore. A very good example that was trending is grip strain.
Okay.
So grip strain is, so first of all, why do scientists and a lot of studies use grip strain? So you can measure grip strain by basically squeezing this apparatus, which is called a dynamometer. And basically, it's very fast to use. If I want to do a study with hundreds of thousands of people, I can go through this assay very easily, and is a very good measure in the general population of maximum strength. What happened is that it is shown to be, you know, correlated with all-cause mortality, correlated with, actually is an independent risk factor for all-cause mortality relative, for example, to the amount of muscle mass you have in your body.
Right.
So it's a very interesting biomarker, very useful. What happened? As soon as people knew, especially biohackers, that grip strength is important, they bought all these grippers and they start using it. And that's not the point. So the point is that it's a good biomarker because if you do, you know, a lot of weight training, for example, it comes as a byproduct of the weight training. That it assumes that the grip strength is a good measure for strength overall in your body. For example, it's highly correlated with leg strength if you measure grip strength and leg strength. Now, if you take grippers and you just train your grip, almost like for arm wrestling, let's say, that's not the point. That it loses its validity as a biomarker. So there are these cases.
Well, correlation is not causation, is what is ultimately. So when you're a biohacker, and in this world that we live in with all these influencers, you basically are going to go try to get your strength stronger just in your grip, just for the purpose of living longer. But it probably makes them sell whatever device that makes your grip stronger. You know, like you can sell a lot of those devices, right? So, well, listen, this has been a very interesting conversation for me, and I really do appreciate all your time. And the company, again, it's NOVOS. And I mean, can you get these products on Amazon, or is it you have to go to the NOVOS site, or how do you get all this stuff?
Yeah, only in novoslabs.com.
Okay, novoslabs.com. Again, that's NOVOS labs.com. And it sounds like, you know, you have like a number of different supplement and also various biological and epigenetic clocks as well. Is there anything else that we ought to know about?
No, I think I'm just working hard to bring more and more. And I basically want to use all the things that are available, you know, to big pharma, biotech.
Yeah.
I want to build all of those techniques to direct to the consumer because I don't understand why they should belong, let's say, to big pharma. So that's what I'm doing.
No, I think that's great. And I think particularly because, you know, the supplement market, especially, it's a wild, wild west. And you just have no idea what you're getting with any of these particular products. So it's nice to have a company that's actually devoted to quality. So I appreciate that. Thanks again for being on, and great talking to you.
Thank you. It was really nice being here. It was really nice meeting you.
Thanks for listening to Sapio with Buck Joffrey. A quick reminder that while I am, in fact, a surgeon, nothing I say should be construed as medical advice. Now, make sure to include your physician in any medical decisions you make. And also, if you're enjoying the show, please make sure to show your support with a like, share, or subscribe. Until next time, this is Buck Joffrey for Sapio with Buck Joffrey.